Okay, here we are. One of those exciting days. Last AMP lecture, right? Thought I would never get here. Do you think we would never get through the book? Yes, okay. Well, so here we are, and today we're going to talk about genetics and heredity and a few other good things. So, as far as genetics go, all of our cells basically have the same genetic information. Every cell will have, for example, the genes that relate to skin color. Every cell will have the genes for making different enzymes, but we only use what's appropriate to that cell. So not every cell uses all of the genetic material that we have. As we talk about genetics, there's a lot of terminology that we have to become familiar with. And so I gave you a list of some things that we're just going to discuss. So start with genetics is the branch of biology that deals with heredity, you know, how inheritance works. And heredity, then when we're talking about heredity, we're talking about the process by which we transfer biological information from parent to offspring. So I think probably everyone knows that when we're talking about this transfer of information, we're talking about chromosomes. And chromosomes are made out of DNA, which we've talked about in the past. And a chromosome will contain, you know, long strands of DNA which coil around. And then within the chromosome, there will be many genes, or sometimes we call them alleles, that contain bits of information. Chromosomes occur normally in pairs, at least in the higher organisms. And in humans, we have 23 pairs of chromosomes for a total of 46 chromosomes. Okay. DNA, deoxyribonucleic acid, we've talked about this a couple of times. It looks like a double helix, has crossbars that are made up of bases, has sides that are made up of sugars and phosphates. Okay. And gene or allele, as I already said, is just a little bit of information on the chromosome that is a bit of information for one thing, for one characteristic, for one protein, for one enzyme, whatever might be the case. Diploid number and haploid number, we were talking about these when we were talking about reproduction. The diploid number is the number of chromosomes that we have in all of our cells except for the reproductive cells. And so again, for humans, this would be 46. And then the haploid number is half that. It's one member of each chromosome pair, so the haploid number for humans would be 23. Any questions? Okay. Some more terminology. When we're talking about pairs of chromosomes, we call the members of a single pair homologous chromosomes. And they're going to have information for the same genetic traits. So if this were a pair of chromosomes, we might have a pair that would have information about eye color, finger length, freckles, or no freckles, hair line, you know, whether your hair line is straight across or comes to a point across, whether your earlobes are attached, and, you know, so on and so forth. So if, say, this was pair 5, then both of these chromosomes would have this kind of information. Now, the information wouldn't necessarily be identical, because one chromosome might have genes for blue eyes and one have genes for dark eyes, brown. One might have genes for having long fingers. The other one might have genes for having short fingers. One might have genes for having freckles. The other one have genes for no freckles. You know, that type of thing. But this is what we're talking about when we're talking about homologous chromosomes. You know, they carry information for the same traits. When we talk about chromosomes, about this 46 chromosomes that we have, these 46 chromosomes that we have are divided into autosomes and sex chromosomes. In humans, we have 2 sex chromosomes, and they make up 1 pair. And then we have 22 pairs of, or 44 autosomes. So the autosomes are the non-sex chromosomes. Typically, the autosomes have a lot more information on them than the sex chromosomes do. We will talk after a while about what happens if we're missing a chromosome. And being short an autosome is much more devastating than being short a sex chromosome. Something else that we've already talked about is meiosis. And meiosis is that type of division where we reduce the chromosome number. The chromosomes replicate, the cytoplasm divides once, half of the chromosomes go to each half, and then the cytoplasm divides again. And so we end up with, in men, 4 sperm cells. In women, we end up with 1 gamete, 1 egg cell, and 3 polar bodies. But we divide the cytoplasm and separate the chromosomes twice, so it reduces the chromosome number. Okay. About what? Okay, when women do meiosis, we get 1 egg cell and 3 polar bodies. Remember that from last time? Okay. One of the things that we didn't talk about when we talked about meiosis, but something that is very essential, is the idea of independent assortment. And that's the idea that when the chromosomes separate, that it's completely at random how they separate. And perhaps this will give you the idea a little bit of doing this. Because here we see 4 examples, okay? This represents a cell that has 6 chromosomes, okay? This represents a cell that has 6 chromosomes, and those chromosomes line up in the middle and they separate. And they can separate so that all the, you know, so that the purple ones are both on this side, the green ones are both on this side, okay? Or they can separate so that we have 2 purple and a green. Here, over here, we have 2 green and a purple. Or they can separate so we have a different green and 2 purple, a different purple and different greens, and so forth. And so you can see that from these same 3 pairs of chromosomes, we can get a lot of variation on how they're going to separate. Is that making sense to you? So if you look at those 4 cells that are being produced in each example, if you think of those as 4 sperm cells, okay. So we talked before about how sometimes characteristics will travel in pairs. And so if this, on this long purple chromosome, just for example, we have the genes for red hair, okay. And on this little crooked chromosome, or bent one, we have the genes for freckles. Okay, if those 2 are traveling together, then we expect the person with red hair and freckles. But look over here. The freckles are over here. You see what I'm saying? When we just have it on, these are the homologous chromosomes. One has genes for freckles, one doesn't. So independent assortment is one of the things that gives us a lot of variety when we're talking about, you know, offspring and what the offspring will look like. You know, sometimes you look at families and you think, where did that person come from? You know, there is so much variation. Other times you see that everybody in the family looks quite a bit alike. But as you look around the world, you can see there's a huge amount of variation, and that's due to independent assortment. Okay. Now, if you want to know what the possibilities are, someone figured this out, that the possibilities are 2 to the nth power when n equals number of pairs. Okay, so in this chromosome, in this organism over here, unnamed, but it has 3 pairs of chromosomes, right? So the possibilities would be 2 to the 3rd. 2 times 2 is 4, times 2 is 8. So we would have 8 different possibilities of what we could end up with in the sperm cell or the egg cell. So for humans, we have 23 pairs. So 2 to the 23rd. Okay, I'm not going to do that one in my head. My head only goes so far. But it's around 8.5 million different possible combinations. Any questions? Okay. Some more terminology. We need to talk about these 2 terms together, genotype and phenotype. Genotype refers to the genetic makeup of an organism. And phenotype refers to the appearance of the organism. In order to make some of this stuff more real, I'm going to give you an example of traits. And I'm going to pick a trait that actually exists in humans, and I'm going to pick a trait that we can actually test for in this classroom. Okay, so I'm going to pick the trait of being able to roll your tongue. Okay, and the being able to roll your tongue, if you know what that means, now this is probably going to be obscene, but nevertheless, it's going like this. Okay, so now you can all go home and say, my teacher stuck her tongue out at me. Well, too bad, life is hard. Okay. The tradition in genetics is that when we're talking about a dominant gene, we use a capital letter. And when we're talking about a recessive gene, we use a small letter. So being able to roll your tongue is a dominant gene. We don't know what dominant means yet, but you'll take my word for it. Okay. So if we let capital T represent a person who can roll their tongue, or the gene for being able to roll your tongue, and small t represent the gene for not being able to roll your tongue, we come to your genotype. Okay. We all have 2 genes for this characteristic. So our genotype could be large t, or it could be small t, or it could be one of each. That would be the 3 genotypes that we could have. Okay. Phenotype refers to how the gene is expressed. And so that's going to be either you can roll your tongue or you can't roll your tongue. All right? So in the privacy of your own mouth, behind your hand if necessary, I want you all to find out if you can roll your tongue. Okay, now, there's no discrimination here, but I just want to know how many people cannot roll their tongue. So we got 1, 2, 3, 4. Okay. So that means everybody else can? Or you were unable to figure it out? Okay. Well, if you cannot roll your tongue, then that is your phenotype. If you cannot roll your tongue, then what is your genotype? Little t, little t. Okay. If you can roll your tongue, what is your genotype? It could either be this one, big T, or big T, little t. Because now we're getting ahead of the story, but we have dominant overshadows recessive. Okay. So we'll do some more things with this. Do not forget whether you can roll your tongue or not. Okay. Two other terms that we need to talk about is homozygous and heterozygous. And homozygous is when both genes in a pair are identical. And then, of course, I think you probably can figure out what heterozygous is. Heterozygous, the two genes in a pair are different. Okay. So looking at this, a person with this genotype, large T, is going to be homozygous for the trait. A person with this genotype, small t, is going to be homozygous for the trait. A person with this genotype is going to be heterozygous for the trait. They're going to possess one of each characteristic. Okay. So here at long last we arrive at dominant and recessive. And dominant genes are those which will show up. Recessive genes are ones which will be hidden if there is a dominant gene, but will be expressed if you're homozygous recessive. Okay. Recessive gene will be hidden if there's a dominant gene. Will show up if you're homozygous recessive. Okay. So right here is here's our homozygous, or excuse me, here's our heterozygous. This person is going to be able to roll their tongue. That's their phenotype. But they carry the recessive gene. They could pass this recessive gene on to their children. All right? So now we come to something called a Punnett square. And a Punnett square is a way of predicting possibilities. Okay. So what you do is we're going to talk about what happens if. Okay. So what happens if a person who is homozygous tongue roller marries a non-roller? All right. So what we do is we put one set of genes across the top, we put the other parent's set of genes across the bottom, and we say this one combines with this one. This one combines with this one. So we fill in the boxes. This gene and this gene. This gene and this gene. And all of the children would be, okay, all of the children would be heterozygous tongue rollers, right? Okay, they would all be able to roll their tongue. Okay. Now, we can use something similar sometimes to figure out possibilities. Okay. My husband cannot roll his tongue. Life is difficult. Other than that, he's almost normal. Okay. So I can roll my tongue. And I happen to know what my genotype is. I know that I'm heterozygous for being able to roll my tongue. Okay. Now, if I knew about my parents, I might be able to figure that out. Okay. But my father's been dead for over 20 years. I have no idea whether he could roll his tongue. My mother's still alive, and I never remember to ask her. I always think about it when I'm giving this lecture. You know, I don't think about it when I'm there. I was there yesterday. Never crossed my mind. Someday I will ask her. But I still know that I'm heterozygous. So how do I know that? This is my genetic makeup. This is my husband's genetic makeup. How do I know this is my genetic makeup? Okay, Jessica, she took your thing. Okay, so we have a child who is a non-roller. Poor kid. But in order to get a child who is a non-roller, they have to get that gene from both parents, right? Okay, so if this is my genetic makeup, looking at our possibilities, we would expect, now this is just telling us possibilities. We would expect that half of our children would have the ability to roll their tongue, but would be heterozygous. And half of our children would not have the ability to roll their tongue. And of course, they're going to be homozygous. And we would have no homozygous rollers, right? Okay. So what are the chances that it's going to work out 50-50? Well, if I were a housefly and I had 12 trillion offspring, we might see that it would work out to be about 50-50. I have 3 children. 2 can roll their tongue, 1 can't. But in order to get this probability to follow through, you do need a lot of offspring. Okay? You can't just take one person. You know, because if you just took my family, you'd say, oh, well, the odds must be 2 to 1, or 1 out of 3 will not be able to roll their tongue, where it's really 50-50. Is that making sense to you? Okay. So you can use Punnett squares to tell you what the probabilities are. Okay. What's the probability of having a boy or a girl if you're pregnant? 50-50, okay? So I have a friend who had 5 boys, was pregnant for the 6th time. What was the probability that she would have another boy? 50-50, right. It's every incident that is the probability. So then why didn't she end up with 3 boys and 3 girls?
The guy's fault.
Pardon? It's the guy's fault. That's a good answer. It was the guy's fault. Okay. But if you took all the children that were born in Indiana during the year 2007, it would probably work out pretty close to being 50-50 boys and girls. Okay. Actually, for some reason that they don't fully understand, for live births, there are usually a few more boys. It doesn't work out quite 50-50. In humans, there's usually a few more males born than females. Of course, they don't last. You know, if you look at statistics, about how many people, like how many people in Indiana are age 1 or age 2 or age 3 or age 4, up until you get to about 19, boys lead the way. And once you get from that 19 to about 25, then it starts to fall off. And there are more women than men. Right? Okay. If you don't believe that, you know, the older you get, the more that's true, that there's more women. You know, if you don't believe that, just think about when you go to church, you know, and you look around at people that are in the age group with your parents, people that are in the age group with your grandparents. Aren't there more women than men? Okay. So there's no known reason for that. My husband and I have conflicting theories about it. My theory is that men just don't hold up, and his theory is that women wear them out. So you have your choice. Okay. Any questions about this? So when we have been talking about Punnett squares and about things like rolling your tongue, we have been talking about dominant and recessive genes. But we also have some other genes that don't follow this pattern. We have what we call codominant and incomplete dominant. And those things sound like we're talking about the same thing, but we're actually not. Okay. In a codominant situation, we have two genes that are both dominant, and they both are fully expressed. Now, you may be wondering how that works. But we have a wonderful example of this in humans, and it is in blood typing. Okay, in blood typing, we have type A. And type A is due to a dominant gene, causes the A antigen to be produced on the erythrocytes. We have type B. And type B is also due to a dominant gene, causes B antigens to be produced on the erythrocytes. We have blood type O, and O is recessive. So if your genotype is AO, your phenotype is A. Right, okay. And then we have type AB. And this is where we see the codominant. Because in type AB, we have the A antigen, and we also have the B antigen on the erythrocytes. So both characteristics are fully expressed. Every erythrocyte will have A and B. Any questions? Okay. In incomplete dominance, we have the phenotype, the appearance, is a compromise. And let me give you an example. Let me give you a non-human example to start with, because I think this illustrates it very well. Okay. And the example I'm going to give you involves a flowering plant called 4 o'clocks. Now, this is not true for all flowering plants, but it is true for this one specific species. Okay. In 4 o'clocks, there is a gene called R that produces red pigment. And so if your genotype is RR, you have red flowers. There is another one. Now, Lindsay, I know you think you know what's coming. There's another gene that is called W for white. And so if your genotype is WW, you have white flowers. Now, have you all figured out where it's going? Okay. If you have one R and one W, your flowers are going to be pink. The phenotype is a compromise. Roan cattle, R-O-A-N, or roan horses. I don't know if you're familiar with either one of those, but that's another example of incomplete dominance in which on the coat of the animal, some hairs will be red and some hairs will be white. Now, it's not going to be patchy like you see with a spotted dog, cow, horse, or whatever. But it's just going to be intermixed. In between the red hairs will be white hairs, or you can say it the other way around, but it's probably usually mostly more red. But some hairs are red, some hairs are white. Now, it doesn't make the horses or the cows look pink, okay? But it does make them look a different shade of red than if all the hairs were the same color. And they call that color roan. Okay. But now for people. Okay. The best example of incomplete dominance in people is sickle cell anemia. If I could spell abnormal, we'd be fine. Okay. Sickle cell anemia is a genetic condition. The dominant gene is for normal hemoglobin. The recessive gene is for abnormal hemoglobin. We looked at this in the lab when we were talking about blood types and blood and so forth. In sickle cell anemia, if you're homozygous for the recessive trait, if you have sickle cell anemia, what will happen is if you're put into a stressful situation or a situation where your oxygen levels become low, then the red blood cells will change shape, and this will make it difficult for them to carry oxygen. They will rupture, your hematocrit will drop, and so forth. Is a life-threatening situation. People die from sickle cell anemia. Okay. In the codominants, what happens is some of the erythrocytes will have normal hemoglobin, and some of the erythrocytes, I'm sorry, I said that wrong. In incomplete dominance, sorry, change that. Okay. In incomplete dominance, some of the cells will have normal hemoglobin, some of the cells will have abnormal hemoglobin. Now, there is no cut and dried rule about what your percentages are. You would think that if your genotype were this, you would have 50% of your cells normal and 50% of your cells abnormal. But that isn't true. Some people have only a small portion of their cells with the abnormal hemoglobin. Some people have a higher than 50% of them with abnormal hemoglobin. So the result is there's a lot of variation. We say that people with this genotype, we say they have the sickle cell trait. We don't say they have sickle cell anemia, we say they have the sickle cell trait. Some people that have this genotype might live a long time, or maybe their entire life, and never be diagnosed. Other people will have some severe crises in their life because their percentage of sickle cell, of abnormal hemoglobin is high enough. Okay. So does this make sense to you? You understand how that works? Okay. One more example and we'll hang it up. Okay. Another example is we have we have genes for hair texture. The H1 is the gene for having straight hair. So if this is your genotype, straight hair. The gene 2 is the gene for having curly hair. So if this is your genetic makeup, you have curly hair. So have you figured out that if you have one of each, this is a codominant situation, you're going to have wavy hair? Okay. Moving along, we have two other situations that are also sound a lot alike, but aren't. And this is multiple alleles. Allele, remember, is the same thing as genes. And polygenes. Okay. Multiple alleles is when we have more than 2 forms of a particular gene. Now, most of what we've talked about so far, we've only had 2 forms of the gene. You could roll your tongue or not roll your tongue. You had straight hair or curly. You had the straight gene or the curly gene. Okay. You had normal hemoglobin or abnormal hemoglobin. Okay. We did talk about one, though, that was multiple alleles, and that was blood typing, which I've erased. Okay. But blood typing, we have A, we have a gene for A, we have a gene for B, and we have a gene for O. And of course, A and B are dominant, O is recessive. But we do have, each person only has 2 genes, but there are 3 possibilities. There's 3 forms of the gene. So does that make sense? Okay. So in order for a person to have type O blood, it's recessive, right? So you're homozygous for that characteristic. Okay. Polygenes is when we have several pairs of alleles that determine a trait. And I have to draw some chromosomes here to show you how this works. Okay, so this is when you have several pairs of genes for the traits. This is going to be things where there's a lot of variation. Look around at the color of your hair. We're making the assumption that nobody has done anything to the color. Okay, all right, now that we've all rolled our eyes. But if you look around and you look at the people in this room who have brown hair, you can see there's a lot of variation in what brown is. You know, there's light brown, dark brown, medium brown, and so forth. Okay. Eye color, skin color, height. You know, we don't have just 2 genes for height. We don't come tall, short, or in the middle. You know, if that were true, then we'd have people that were 6 foot tall, people that were 5'8", and people that were 5'4", and there would be no variation. But we have a lot of variation in height. Okay. So let me show you how this can work and how we can get a lot of variation among our offspring from the same set of parents. So looking at these chromosomes, I have to tell you a little story about Tommy and Susie. Okay, and so this is going to be Tommy, and this is going to be Susie. And they both were of average height. Okay. And I forget what average height is, but in the United States, I think average height for men is like 5'9" or something like that, and for women maybe 5'5", 5'6". Okay, so they were both average height. And after hanging around in A&P class and lab and all of this good stuff, they decide to get married. And then after a little bit of time, they decide that they're going to have a child. So here we go. They're going to have a child. So one of these chromosomes is going to end up in the egg, and one of them is going to end up in the sperm. So let's just, for random selection, independent assortment, and all that good stuff, these are going to be the chromosomes that are in that baby number 1 gets. Okay. Now, all during this pregnancy, Tommy had been talking about his dreams for somebody that would have a career in the NBA or the WNBA. So one of the things that's very helpful for having a career in that sport is being tall. Okay, now I forgot to tell you something. The circles that are filled in represent tall. And the empty circles represent short. Okay. And six genes on each chromosome. Tommy has 6 talls and 6 shorts. Susie has 6 talls and 6 shorts. But look at baby. What do you think? Baby is going to be kind of short. You know, out of a possible 12 genes for tallness, baby only got 2. So maybe baby's going to have to be a cheerleader. Maybe baby can be a NASCAR driver. You know, tallness is not a help in getting in those cars. Okay. So that's okay. Tommy's satisfied. He likes baby, whatever. So let's have another baby. And this will be baby number 2. So how many genes for tallness did baby number 2 get? 10 out of a possible 12, right? Okay, so now we have this kid, you know, and everybody's going, oh, gee, shall we look at the UPS driver? I mean, no, it used to be the milkman, now it's the UPS driver. Okay, so what do you think? Can 2 average height adults have a child that's 6'6"? Sure can. Known them to do it. Okay. And so if we wanted to carry this further, we could have baby number 3, and baby number 3 could get this chromosome and this chromosome, and we'd have a child that would be average height. So assuming that these children were all of the same gender, you're going to have a huge variation in their appearance. Now, you do realize that I set you up for this. Because when I distributed the 6 chromosomes for tallness, I put 5 of them on one chromosome. And the same over there with Susie. You know, so I did set this up. It wouldn't necessarily work out that you would have 5 on one chromosome. But if you did, this is how you could get a child that was much taller or much shorter than their parents and their siblings. Same thing's going to go for skin color, hair color, eye color, several things. Any questions about that? Okay. The next slide shows us this possibility, and it's showing us, you know, the proportion of offspring and how they will vary in terms of height. This could work for height. This could work for skin color, which I guess is what I have printed up there. All sorts of things where we have polygenes. Okay. Any questions? The next thing we're going to talk about is sex-linked and sex-influenced characteristics. And we'll do sex-linked first. Sex-linked characteristics are genes that are carried on the X chromosome. Okay. Most of these characteristics have been found to be recessive. And so what this means is this is something that is much more apt to show up in a man than it is in a woman. Because since it's sex-linked and it's on the X chromosome and it's recessive, in order to get it to show up in females, you need to have 2 recessive genes. So one example of this, something that is sex-linked, is color blindness. Another one is hemophilia. Hemophilia, of course, is when your blood doesn't clot. Another one is a particular form of muscular dystrophy called Duchenne muscular dystrophy. So let's talk a little bit about how this inheritance works. And this is an easy one for me to do because my father-in-law, who is now deceased, but he was colorblind. And so, since this is a disease that shows up in men, fortunately or unfortunately, as the case may be, he had 2 children, both boys. He had 4 grandchildren, all male. Of those grandchildren, 3 of them are my children. And so, of his 4 grandchildren and his 2 sons, how many of those do you think are colorblind? Okay, how many of you think all 6 are? Put your hand up. 5, 4. Nobody can remember how to raise their hand. Let's go with 0. 1, 2, 3, 4. Okay, well, let's find out. We'll do a Punnett square. Now this is on, okay, we are going to use, this is always a difficult one. We're going to use large C for normal, and we're going to use the small c that has this little tail on it for colorblind. We got that one? Okay, so this is my father-in-law. He's carrying the gene for colorblindness. This is my mother-in-law. In terms of vision, completely normal. We won't go any further. Okay. I can imagine what my daughter-in-law is saying. Okay, so let's look at the possibilities. So here's their probabilities, possibilities, whichever way you want to look at it. Girls. This girl would be a carrier for colorblindness, right? Okay, she got the colorblindness gene from her father. She got the normal gene from her mother. Colorblindness is recessive. Okay, this girl would also be a carrier for colorblindness but have normal vision. So had they had daughters, they would have been carriers for the gene, right? 100%. Okay, they didn't have any daughters. They had these 2 sons. Where's the colorblindness gene? In order to be a boy, you get the Y from your father, right? And the colorblindness gene is on the X. You got the X from your mother. You see how that works? So the answer is none of the sons were colorblind. None of the grandsons are colorblind. Okay. So now my question is, where did my father-in-law get this gene? Who are we going to blame? His mother, his dad? We're going to blame his mother, right? He got a Y chromosome from his dad. Only possibility if he's a boy. So he got this from his mother. And nobody remembers whether she was colorblind or whether she was a carrier, because she died when the oldest child was 12, and it's just one of those things that nobody knows. She could have been either one. She could have been colorblind. She could have been a carrier for colorblindness. Okay, any questions? Now, my father-in-law's been dead a long time, but he has 2 sisters. So, and he had 4 brothers. No, 3 brothers. Okay, but he has sisters. So let's look at his mother. Let's just say she was a carrier. If this is my husband's grandmother, okay, we lost a girl along here someplace, right there. Okay. If this was my husband's grandmother, then you can see for the girls, they would have been carriers. Or they would have been, no, sorry, for the girls, they would have been 50% chance of being a carrier, 50% chance of being normal. You see how that works? She had one normal gene, we're making her a carrier. She had one normal gene, one colorblind gene, okay? For the boys, 50-50 that they would be colorblind. Okay. Of my husband's aunts, one never had any children. So we don't know whether she's a carrier or not. But one did. And so this is Aunt Ellen. And Aunt Ellen proved to be a carrier for colorblindness. Okay. Now, if she and her husband had had any boys, there would have been a 50-50 chance that they would have been colorblind, right? They didn't have any sons. All they had was girls. But there's one little detail I forgot to tell you about the man she married. Okay, so let's look at the possibilities for girls. Okay, 50-50 chance that the girls would be carriers, the other 50 chance that they would be colorblind. Okay, they had 4 children, all girls. What do you think they turned out with? Well, 50-50 chance, you could expect that they might be 2 and 2. They're 4 and 0. All of the girls are colorblind. So in that branch of the Calvert family, that gene is alive and well. Because here we have these 4 colorblind women, 3 of whom have reproduced, 2 of whom, let's see, I have to think, 2? Yeah, 2 of whom have grandchildren. And so, not only children, but grandchildren. And so we don't see these people a lot, maybe once or twice a year, but when we do get together and some of the grandchildren and great-grandchildren have started to go to school and children have started to go to school, strangest thing happens when those little boys go to kindergarten screening. Their mothers are colorblind. What chance do little boys have? You know, they're all colorblind. All of the sons of these girls were colorblind. There was no other possibility. So Aunt Ellen and I have our laughs. She'll come up and she'll say, oh, you know, this one started in kindergarten, or this one went to pre-kindergarten screening, and guess what they told him? And we're always able to guess. Okay. Any questions? You see how sex-linked inheritance works. Now, being colorblind is usually not a life-threatening situation, especially since they've standardized stoplights and red's always on top and green's always on the bottom, which wasn't always the case. I'm not real sure I'd want a nurse passing meds who was severely colorblind. Okay. But, you know, for most of the part, that's not life-threatening. Having hemophilia is. You know, hemophilia works exactly the same way. The adherence patterns are exactly the same. This is a life-threatening condition. This used to be a lethal condition for females. So when you would have looked at this, you know, looked at these different situations, if you were homozygous for the gene for hemophilia and you were female, you were going to die when you reached puberty. just no way around it. Now we have medical treatment for that, we can give them clotting factors and this type of thing. Okay. Any questions? Okay. Sex influence characteristics. Sex influence characteristics involve genes that can be on any of the chromosomes, but how the genes behave depends upon what sex hormones are present. And so the one that we probably hear the most about is male pattern baldness. And the gene for male pattern baldness is recessive in females that are producing estrogen. So if you're going to be bald and female, you have to be homozygous for the characteristic. You have to have both genes. For men, it is dominant in the presence of testosterone. So if men are going to have one copy of the gene, one of the genes, they're going to probably become bald at a fairly early age because it's dominant in the presence of testosterone, recessive in the presence of estrogen. Okay. Now there is another trait that's not nearly as well publicized that is also sex influenced, and it has to do with the length of your index finger. Okay, and this gene is just the reverse. It's dominant in women, recessive in men. And the gene makes your index finger equal to or longer than your ring finger. So look at your index finger, look at your ring finger, and if your index finger is as long or longer than your ring finger, you possess this gene. And so who cares? I hate to say this, but men wear a lot of baseball caps. You don't see women wearing gloves. We're not ashamed of having fingers that are strangely linked, have strange links. Okay, moving on. Something else that contributes to genetic variety are mutations. And mutations are changes that happen in the genetic makeup. And we classify them according to where they're located. If they're located in our reproductive organs, and by this I'm saying in the ovary or in the testes, then we call them germ mutations. And germ mutations can be passed on to the next generation. So somatic mutations are mutations that occur anyplace else other than in the ovary or the testes. And these aren't going to be passed on to the next generation. So what are some of the things that cause mutations? Pardon?
Cancer? Well, they are.
Cancer is a mutation, yeah, okay. What are some of the things that cause mutations? Exposure to radiation, right? Okay. This is why when you go to the dentist office, you go to the to an X-ray place, they have signs up. If you think you might be pregnant, let us know. Even if they don't think you're going to be pregnant, they will bring out the little lead apron and drape it over you. Do they care about your hands and your feet? No, they drape it over your reproductive parts, right? Okay, because they don't want to have a germ mutation. Some chemicals will cause mutations. Sometimes you'll see industrial strength cleaners that will cause mutations. They'll say right on it, mutagenic agent. Do not get this on your skin. Agent Orange. You're way too young to remember Agent Orange, but Agent Orange was a chemical that was sprayed on the jungles during the Vietnam War. And the idea was it would make the foliage fall off the leaves fall off of the trees. And so snipers were hiding in the jungles. This was the answer to that problem. But it was also a mutagenic agent, at least according to everybody except the United States government. But we do know that people who were in the jungle when the jungle was sprayed ended up with lots and lots of mutations. And some of them were germ mutations as well as somatic mutations. Okay. We take a lot of precautions. We not only have the lead aprons, but sometimes when there's a lot of solar radiation, when every so often there will be a period of high solar flare activity, and they will sometimes limit how high airplanes can fly or what particular paths airplanes can take if they are flying very high. And then about 15 or 20 years ago, we had that event called Chernobyl. And again, you're probably too young to remember that, but it was when the atomic reactor blew up in Russia. And for a huge area around this, there was lots and lots of mutations, not only among the people, but also among the plants, among the wildlife. And they're still seeing the results of this. You know, they're still seeing an abnormally large number of mutations occurring among organisms that were exposed to that. Sometimes mutations are good. We have a tendency to think that mutations are bad, and sometimes mutations are actually lethal because, as Heather said a little bit ago, you know, cancer is a mutation in the cell. We think of mutations as being bad, you know, things that are going to be harmful to us, but we undoubtedly have many, many mutations that we are completely unaware of, maybe things that have made us more successful. You know, if you would have a mutation, let's say, for example, you're a rabbit, and you have a mutation in your legs that makes it possible for you to run just a little bit faster. You know, so that means you're going to be able to get away from your predators. Not only is it going to make you more successful, but you probably are going to be the rabbit that survives to reproduce. And so undoubtedly, in the past, we've had mutations that have become the normal in populations because they've been something that has made the organism more successful. But they equally could be something that would make us less successful. Okay. Anyway, they do contribute to genetic variety. Something else that contributes to genetic variety is crossing over. And this is something that undoubtedly has happened in the past and probably is still continuing to happen in the past. We usually think in people that if you have a person that has blue eyes, they will probably have light hair. And if you have a person that has very dark eyes, they probably have brown or black hair. So we're working on the theory that hair color and eye color genes are on the same chromosomes. And so what we've done here is we have let the capital letters represent the darker brown hair versus blonde hair. We've let the capital E's represent brown eyes versus blue eyes. And so probably what happened, you know, generations in the past was the brown hair and the brown eyes were on the same chromosome. The blonde hair and the blue eyes were on the same chromosome. But during mitosis, when the chromosomes come together in the center of the cell, some of them probably wrapped around each other. And then what happened was that when they wrapped around each other, they broke and refused. And so now we have a chromosome that has brown eyes and blonde hair. We have a chromosome that has blue eyes and brown hair. And so once that happened, we now have we now have more variation. And so it's no longer a hard and fast rule that those characteristics always go together. But this is something that happens all the time, just as a normal, I guess we'll say a normal mistake, or just as something that can occur. And most of the time it has no consequence other than it adds to the variation. Any questions about that? Okay. Let's go talk about something. Let's go ahead and talk about something that is a little bit more serious, and that is non-disjunction. And as we talk about that, let's back up and look at what's happening here. Okay. What we're seeing here is we're seeing where chromosomes have replicated. You remember when we're talking about mitosis? At the end of interphase, the chromosomes replicate. So here we see one pair of chromosomes, they have replicated. Now, as time goes by, what will happen is these chromosomes will spread apart, and one of these will go to one cell, and one member of each pair will go to the other cell when we're doing mitosis. So we'll end up with a cell that has, each cell has two copies of or has a copy of the original two chromosomes. In non-disjunction, the separation fails to occur. The chromatids that are holding one pair together don't separate. And so with non-disjunction, what we end up with is we end up with one cell that has 47 chromosomes and one cell that has 45 chromosomes. And we call this trisomy. Okay. Trisomy is when you have the extra chromosome. Instead of having two members of a pair, you have the third chromosome. So when we talk about this, we say trisomy 5. That means you have an extra fifth. Trisomy 21, you have an extra chromosome of pair number 21. Okay. Now, you may be familiar with this and know that trisomy 1 is what we call Down syndrome, in which there is an extra 21st chromosome. And this is an abnormal situation, and it's going to cause abnormal development. Now, there's not a lot of trisomies that we see. We see 22. 22 is very similar to Downs, except the skeletal deformities are quite a bit more serious. Trisomy 13, 15, and 18. These are very severe defects. Life expectancy is 1 year or less. Although I actually have a student, I've had a student recently who had a child with trisomy 18 who was, I believe, 6 years old. But lots of problems. I mean, this is a child that basically has to have everything done for them. The other trisomies seem to be lethal, except for these 5, 13, 15, 18, 21, and 22, they seem to be a lethal situation, and so there is not a live birth. That there's either a spontaneous miscarriage or the child is stillborn. That it's so wrong. Okay. The other cell is going to be shorter chromosome, and this, of course, is usually lethal. Now, there is one exception to this when it's not lethal, but usually this is lethal. Okay. The way this originates is most of the time it happens during gamete formation. So we get a gamete that has 23 chromosomes, a gamete that has 24 chromosomes, and then we get the diploid number that is wrong. Okay. I'm not going to ask you to remember, which trisomy is which, except you probably need to know that 21 is Down syndrome. This is just simply a picture showing non-disjunction. This actually happens to be non-disjunction involving sex chromosomes. Now, the trisomies that we talked about were ones that involved autosomes, but this is showing you what happens with sex chromosomes. So here we have where we have an extra X in one egg cell, or this egg cell doesn't have an X, we can have a sperm cell that, and then fertilized with a normal sperm cell. We're just showing the non-disjunction happening here with the female. We're going to talk about what happens in these different cases as far as what goes when we have abnormal sex chromosomes. Okay. But before we do that, okay, we're going to talk a little bit about some genetic diseases. And I have listed here a few genetic diseases. I would like for you to have just a general understanding of these. We're not going to get into great detail about them, but these are all things that are genetic. Okay. The first one is cystic fibrosis. Cystic fibrosis is the result of a recessive gene. The problem with cystic fibrosis is that this abnormal gene causes you to produce a very thick mucus in your respiratory tract and your digestive tract. As a result of that, it's going to be difficult for you to breathe. These children are going to have a lot of what we might call phlegm, a lot of mucus in their respiratory tract and their lungs. That's going to have to be removed. The mucus in their digestive tract will make it difficult for them to absorb their food. This occurs in 2,000 births. And life expectancy used to be about 20 years old. Actually, today, life expectancy is about 40. It may be longer than that. We just know that we have people who have reached the age of 40 that have cystic fibrosis. So we don't know how much longer they will be able to live. But it's because we've made a lot of strides in treatment. There is no cure for this disease. You can do genetic testing for this to find out whether or not you're a carrier. So, if you're a carrier for cystic fibrosis and your spouse is a carrier for cystic fibrosis, this is when you're going to have the problem. Okay. Tay-Sachs is also a recessive gene. This is a very nasty disease. Life expectancy is about 3 years. Because of a mutation, they cannot make an enzyme that is needed to break down a lipid that will be found in the nervous system. As a result of this, there's a lot of damage to the nervous system. And so what will happen is a child with Tay-Sachs will probably develop normally until they're about 6 months old, and then they will start to regress. And it's not nice. I mean, they regress to the point that they cannot feed themselves. Many times they lose their sight, and life expectancy is very short. Okay. Sickle cell anemia. We have talked about sickle cell anemia. This, of course, works by incomplete dominance. And so you can have the condition, sickle cell anemia, or you can have one normal gene, one abnormal gene in which you have sickle cell trait. Okay. PKU, phenylketonuria. Does anybody have diet pop? Of course, I brought coffee today. In this particular disease, you lack the enzyme to break down phenylalanine. it will accumulate and damage your nervous system. This is something that newborn babies are tested for immediately. There is no cure for this, but we really can keep the damage from happening if we're aware of the fact that the child has PKU. Because what happens is you simply need to eliminate that material from your diet. And that's why I said, does anybody have diet pop? Because the sweetener in diet pop contains phenylalanine, and on the can of diet pop, it will have this in red letters. You know, it'll say Cochin phytoketonuria. This contains phenylalanine. Other packaged foods or other processed foods that you buy will be clearly labeled that this substance is in it. If you can avoid this substance, you basically can develop normally. If you do not avoid it, you will be mentally retarded. And it is damage that is not reversible. So this is why it's important that the child be tested right away. And so this is something that they test very quickly. Okay. A rather rare condition, but one that I will mention, is albinism. And albinism, which not only occurs in humans but also occurs in most vertebrate animals, is when the person lacks most or all pigment. And so this presents some problems in terms of exposure to sun. It presents some problems in terms of damage to their eyes. It is a recessive condition. Any questions? Okay. Here are 3 conditions that are dominant. And they are autosomal dominant genes, which means they're not found on the sex chromosomes, but rather on the autosomal chromosomes. And we'll start with the bottom one, because this is where we have the picture. But achondroplasia is dwarfism. And being an autosomal dominant gene, this means if one parent possesses the gene, it will be passed on to, there will be a 50% chance of passing it on to your offspring. Okay. They think that in most cases, achondroplasia is the result of a spontaneous mutation, because many times people who do not carry this gene will have a child with achondroplasia. And so that it is happening as a spontaneous mutation. Of course, if both parents have the gene, well, then you're going to expect that it will pass to the offspring. Okay. Marfan syndrome. Marfan syndrome is a dominant gene. A person who possesses this gene is usually tall, thin, has long arms, long legs, long fingers, an NBA, answer to an NBA coach's dream. You know, they have the build for a basketball player. They have a tendency to be nearsighted, but that's okay, you can take care of that. The problem is they also usually have a weak wall in their aorta. And so this is something that, if it's not repaired, can be obviously fatal. And we've had a couple of instances in the last 10 years in which athletes have died because either they were diagnosed and didn't follow instructions, or they were not diagnosed with the condition. But it is a dominant gene, 50% chance that you will pass it to your offspring if one parent has it. Okay. By far the worst of these 3 is Huntington's disease. Huntington's disease is a neurological disorder. And one of the really bad things about it is it doesn't usually show up until middle age. In the past, when we didn't understand how this worked, or people didn't understand what what disease their parents possessed, many times they would have reproduced before they were aware they had the condition. If you have Huntington's disease, you have a 50% chance that you will pass it to your offspring. The person starts out having balance and coordination problems, symptoms similar to Parkinson's disease, but unfortunately, it progresses, and it's not only a physical deterioration, but it is also a mental deterioration. Sort of a nightmare between Alzheimer's and Parkinson's. Okay. There is testing for this condition. Usually you have to, many times you have to undergo psychological testing before your family doctor will let you have the test to find out whether or not you have Huntington's disease. If you were 19 or 20 years old and you've suddenly found out that one of your parents had this disease, would you want to be tested? Would you want to know that this was going to be your fate in probably another 20, 25 years? If you were thinking about having children, you might want to be tested so you would know whether it was safe to have them or not. But then how would you feel if you were tested and you were fine? You had not gotten the gene, but your sibling had. You know what survivor guilt is? It might be, something that would be, it's definitely not one of our nicer gene or diseases. Any questions? Okay. So, as promised, let's go ahead and talk about what happens when we have an abnormal number of sex chromosomes. And I put the conditions on here, but I'm going to go ahead and go past this and show you again the non-disjunction happening and what the possibilities are. Okay. So let's start down here in the lower right side with the person who is XXY. Now, you remember when we talked about the fact that if you have the X chromosome in the fetus is going to become male, that we became female by default. Well, just to empower the men in the room, I want to tell you that if you have two X's and a Y, you still are going to become male. That Y chromosome is pretty strong. So people who have Klinefelter syndrome, in which their genetic makeup is XXY, will be identified as male. They tend to be borderline on the low side of average intelligence. Often they are sterile, but to the world, you're probably not going to be able to identify this person just based on phenotype. This is somebody who's probably going to function in society as a male. Nobody's going to be the wiser. Okay. Going up, we have XXX, or sometimes they call that super female, in which the person has 3 X chromosomes. This person, again, will be fairly normal, probably totally unaware of this, other than she may have trouble reproducing. Some people with this genetic makeup are able to reproduce, some aren't. Okay, but functionally is going to be fairly normal. Okay. Going over to the left side, if you have a person that has a single X, that person will be identified as female. We call this Turner syndrome. Okay. Remember a while ago that I said, usually if you're short a chromosome, an entire chromosome, this is not going to be a viable offspring. The person's not going to live. This is the exception. And this exception happens because there's a lot less genetic material on the sex chromosomes than there is on the autosomes. So you can actually survive with just a single X chromosome and not have the second chromosome. Okay. This person is going to tend to be short. Usually they lack the secondary sex characteristics. Intellect may or may not be affected. Some people are extremely intelligent with this condition. And a lot of times when a person is identified with Turner's syndrome, they will give them female hormones to cause them to develop secondary sex characteristics. Very good chance that they will be sterile. Okay. And then we come down to the lower left, and if all you end up with is a Y, it's not viable. Okay, a Y has less genetic material than the X, and so that person is not going to develop. Any questions? Now, there is another possibility. If non-disjunction takes place in the male rather than the female, then what we could end up with is we could end up with XYY, in which we have two Y chromosomes. Okay. This person, XYY, this person tends to be extra tall, and of course is male. Okay. Extra tall. Normal to low normal intellect. And there's kind of an interesting little story that goes with this genetic makeup. Back in the 1960s, a long time ago, there was a researcher in England who was researching who knows what. But anyway, he was he had permission from the penal system, and he was going around to prisons and asking inmates for a blood sample so that they could do chromosomal studies. And after doing these studies, he noticed that in the male population that was in prison, compared to the male population that wasn't in prison, there was a much higher incidence of this particular genetic makeup of the XYY. And so based on just that bit of information, he wrote a couple of papers, held a press conference, did all the things that they do, and said, hey, you know what? These people shouldn't be in prison. It's not their fault. It's their genetic makeup. You know, having that extra Y chromosome means they produce extra testosterone, and they're going to be more aggressive. And so it really isn't their fault. Well, he really didn't get very many people that agreed with him. In fact, he kind of got laughed out of science, so to speak, and it's just kind of an interesting story. But once in a while, you will still see some defense attorney who goes back, finds this work, and offers this as a defense. You know, it's really not his fault, Your Honor. You know, genetically, he is XYY, and the whole blah, 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 blah. So it's one of those things. Makes genetics kind of interesting sometimes. And then, of course, we have the urban legends. And the urban legends say that Charles Manson and Richard Speck were both XYY. Okay. Charles Manson refuses to be tested. Richard Speck is dead, and so we'll never know. Okay. Do you know who those people are? Good. Okay. Any questions? Okay. That brings us to fetal testing. We can do amniocentesis. We can also do either a sample of taking out a sample of amniotic fluid, or the newer thing that they do is they go in and they draw off a sample of the chorionic villi. They're actually taking very early in development, a small sample of that, and testing to see if there's any problems with the offspring. Okay. Any questions? Okay, we're finished.